The Bloom syndrome complex participates in a specific process of DNA repair, termed homologous recombination. In this figure, we highlight how homologous recombination is required to repair double-strand breaks in DNA. In the first stage, called presynapsis, the 3' strand of the broken DNA end is converted to single-stranded DNA via the concerted exonuclease action of the mR11, CTIP, XO1 and DNA2 nucleases. This process is known as resection. Resected DNA is then coated with a filament of Rad51 protein in a process promoted by BRCA1 and BRCA2. The Rad51 filament stimulates invasion of the resected DNA into a homologous template, creating a displacement loop, or a D loop. This second stage is called synapsis. Several proteins are involved in stabilizing the D loop in vitro to promote synapsis, including Rad54 and Rad51 paralogs. The third stage, post-synapsis, has multiple outcomes. In the outcome of synthesis-dependent strand annealing, short regions of newly synthesized DNA are displaced from the template strand and annealed, then ligated to the resected DNA that comes from the other side of the double-strand break, or the second end. In a different outcome, the second end is instead annealed to the displaced strand of the D-loop, creating an additional substrate for polymerase action. Complete DNA synthesis leads to the formation of a structure called the double holiday junction, which topologically links the two parental DNA molecules. A third outcome, termed break-induced replication, occurs when the repaired end does not have a corresponding downstream sequence with which to anneal, for example when the original break was located at a replication fork. DNA synthesis can occur indefinitely towards the next origin or the end of the chromosome. Homologous recombination is typically an error-free repair process. However, its fidelity is dependent upon the Bloom syndrome complex. During presynapsis, the Bloom syndrome complex promotes resection through a direct interaction with DNA2 and XO1 to enlarge the size of the single-stranded DNA Rad51 filament available for homology search. In contrast, during synapsis, the Bloom syndrome complex can evict an invading single-stranded DNA strand from a D-loop when the sequence being invaded does not fully match. Or alternatively, it can stabilize Rad51-coated, fully homologous D-loops. Post-synapsis, the Bloom syndrome complex can stimulate break-induced replication in the absence of a second end by promoting DNA polymerase delta extension of the D-loop, or it can also promote capture of the second end if one is available. Finally, the Bloom syndrome complex is the only enzyme capable of untangling the double holiday junction structure without a risk of crossovers occurring. In Bloom syndrome, when the Bloom syndrome complex is absent, homologous recombination occurs with reduced fidelity regulation, decreased resection and increased crossing over, with the consequences being recombination-mediated insertions, deletions and chromosome rearrangements, leading to cancer and other severe phenotypes. Fidelity regulation by the Bloom syndrome complex is therefore critical to all aspects of homologous recombination.